I'm here with Thomas Rasmussen, who's the Associate Director of HIV Cure Clinical Trials at Doherty, and both of us have just come back from Mexico at the IAS 2019 Science Meeting, and are going to talk a little bit about what our learning was in relationship to HIV cure research. And I thought I'd start by briefly talking about the workshop that was held before the main meeting. At every IAS science meeting, the IAS now holds a single topic workshop on an area that's highly relevant to HIV cure research, and this year the topic was HIV and hepatitis B cure research. And for those of you that don't know much about hepatitis B, it's very similar to HIV in that it requires long-term antiviral treatment, and antivirals generally don't cure hepatitis B. The big difference to HIV, though, is that a percentage of people are cured by antiviral drugs, and the hepatitis B community can also predict who is cured by detecting antibodies to a part of the hepatitis B virus. So there's lots of similarities but also some differences to the work that we do in HIV cure, and the workshop did bring together the two fields, and I thought it was fascinating. I have always worked on those two viruses, but many others who have never done any work in hepatitis B were also really fascinated by the differences and also the strong similarities. So just very briefly, a few key differences are that hepatitis B could potentially be cured by antivirals alone. The reason why you can't cure hepatitis B is due to something called cccDNA. So we heard lots about new antivirals that may potentially get rid of the cccDNA. The other approach that's very similar between HIV and Hep B cure is that there's a strong interest in inducing immunity to the virus by either by boosting the immune system in three ways, and these were beautifully summarised by a talk from Marla Maini from University College London, I think so, who described the strategies for altering the immune response to hepatitis B as revving the engine, rejuvenating the immune system, or replacing the immune system. And she used a great analogy to an old car, so that you might be revving up whatever is there, with an example of that approach is TLR agonists. You might rejuvenate whatever's there, an example is immune checkpoint blockers, or you might replace the immune system with something like CAR T cells. And all of those strategies are being tested in HIV and also being tested in hepatitis B. And finally, one of the key differences, I think, between the two fields is that hepatitis B cure has become an area of intense investigation, not just by academics, but by many companies. So lots of small pharmaceutical companies and large pharma have active programs in hepatitis B. So we talked a lot about why there's that difference. In HIV, there are some big pharma companies active in the field, but it's not as an attractive area for small pharma companies, and I think that's likely related to the fact that some people can be cured of hepatitis B currently, and that there's a biomarker for cure. So it was a really interesting couple of days, and I think the two fields learnt a lot from each other, and I hope we can do a lot more together. And finally, we co-hosted that meeting with ICE-HBV, the International Coalition for the Eradication of Hepatitis B, which is based here at the Doherty, and also partners with ANRF's German funders and the Hepatitis B Foundation. And so I hope we can do or think about some ongoing activities with ICE-HBV in relation to some of the issues that were discussed at the workshop. Now, there were a few interesting primary presentations also at the workshop, which were then presented at the main meeting. And Thomas, I think, went to a lot of the main meeting, I think much more than I was able to do. And Thomas, do you want to just briefly tell us about what the highlights were for you in some of the work that you saw presented?
Yes, surely. One of the studies that I've been looking forward to hearing about going to the conference was actually a study of a compound that's been initially investigated in hepatitis B and now more refocused towards HIV cure context. So this is a compound developed by Gilead, and it's a TLR7 agonist. So their clinical study in HIV was presented at a cure workshop, a cure pre-conference workshop, and again at the main conference. And there's been a big interest in this molecule because a few studies in non-human primates in the last 3-4 years have shown, one, that this compound can activate latent virus such that the immune system can see infected cells, but also secondly, this may be a quite good approach to enhancing antiviral immunity both in the innate and adaptive immune system. So there was quite a bit of interest in that clinical trial, which was presented at the conference, and they performed this trial such that they looked at 6 different doses of the TLR7 agonist and compared this to a placebo group. And just very briefly, what they found was that the TLR7 agonist, when you used higher doses, was still quite safe, and it could activate some of the immune effector subsets that you would expect have a role in fighting infected cells, including NK cells and CD4 T cells. However, the virology data showed that there was no activation of latent virus, so it doesn't appear to be having the same effect during human infection as compared to the SIV infection in monkeys in terms of reversing latent HIV. I think that was an important take-home message, and overall the conclusion with that compound is probably that it's well suited for combination approaches where you might combine this with a vaccine or with antibodies, but on its own it's probably unlikely to play a major role in eliminating HIV-infected cells. So I think that was one important study for the conference. Another thing I found quite interesting, this was sort of packaged into a symposium session, so not an all abstract, but researchers from the Doherty Institute in Boston presented some data on a patient that's actually been followed in San Francisco, now termed San Francisco patient. So this is a guy who is what is we call an elite controller, so he controls virus without any antiretroviral therapy. But in this case, they term this exceptional elite control because it's so effective. The patient was diagnosed with HIV back in '92 and has been off treatment ever since. And in that period, since 1992, they have 34 viral load measurements, all undetectable. And more recently, they looked at his cells to see if they could find any signs of intact virus. So they actually analyzed 1.5 billion PBMCs to look for virus, and all they found was one intact or nearly intact HIV virus, but it contained hypermutations, so definitely not very functional. And then they found 15 sequences that had large deletions. So they proposed sort of the slight provocative hypothesis that this patient might have been maybe not cured, but maybe have cleared his own virus through very exceptional T-cell control of infected cells. So I think that's posing an idea that through very potent CD8 T-cell control of the infection, that this could, in a few individuals, lead to a cure, which could inform, I think, therapeutic approaches to boosting T-cell function in the future.
Did they show CD8 T-cell activity in the presentation?
From memory, no.
Because the other interpretation could be that if you could cripple the virus enough by mutating it enough, it will stop it coming back. And so I think just to explain that we now know that of all the virus that hangs around in people on treatment, most of it's actually what we call defective, meaning it's got big gaps in the virus, or the virus is mutated and can't grow on its own, and very few of the viruses left in people on treatment is intact. So I wonder whether another interpretation is that if this man is a true cure, that if you cripple the virus enough or mutate everything that's there, you could stop virus coming back. We just don't know how to do that at the moment, and it could be the immune system, but there could be other ways to do that.
True. The other thing that I think was kind of interesting also in a cure context is the long-term data coming out on some of the antiretrovirals, especially dolutegravir and this new compound TAF, that in large clinical trials show that there's actually progressive weight gain, especially among women with those compounds. This is something that's currently been looked into much more, but it's just showing that although we anticipate newer compounds are becoming better and better, there may still be long-term toxicities that could emphasize also the need for a cure.
Yeah, I thought that was fascinating because we've sort of seen this before, that you, when you develop a drug, it goes through phase 1, 2, 3 testing, and it's only when it's really out in clinical practice that sometimes you observe adverse events that weren't identified in the clinical trials.
And you have that longer follow-up too.
Yeah, that's sort of the history of lipodystrophy, for example, the early anti-HIV drugs. We still don't really understand what's causing the weight gain, but it's fascinating to me that we're only just beginning to uncover whether it's dolutegravir or the combination of dolutegravir and TAF. And yes, as you say, if there is these long-term toxicities, it does argue a greater need that we need some way to allow people to stop antiviral therapy.
Yes.
Just very briefly, we had quite a good and strong contingent from the Doherty at the meeting. Thomas gave an invited talk on latency reversing agents. Yuri Simons presented some work that he's doing with UCSF, understanding how virus persists on treatment through what we call clonal expansion or cell division, which allows large pools of identical virus, and Yuri showed that this is more common in some cells rather than others. Jen Zabato and Jen Bordsley were both there presenting their work on co-infection, which is highly relevant, HIV, hepatitis B co-infection, highly relevant to the cure workshop. And Jared Stern, one of our PhD students, presented his work at an oral poster session on looking at factors that control HIV production in people on treatment. And he had very interesting work on whether HIV production has a circadian rhythm. So we know that lots of different bodily functions vary over a 24-hour period, and we think control of HIV may also vary over that time. Finally, Thomas, IAS often puts a lot of emphasis on community engagement, which is absolutely appropriate and has been the hallmark of the HIV response from the beginning. What did you learn about community response to HIV cure research while you were in Mexico?
Well, I think one of the sort of the really interesting things about this conference and the tradition it has around community participation is the intermingling scientific presentations with presentations from key community members, which is creating a different shape of the entire session, and reminding us all that we're speaking to a broader audience and we have interests coming from different angles, which are ultimately all around creating better treatment opportunities and understanding the virus better.
Yeah, and actually that was another issue with the HPV cure discussion, is that HIV has been characterised by this very strong voice of community in everything that's been done, and hepatitis B has not had that strong community voice, or something they're very keen to strengthen and learn from the HIV context. Well, thanks very much. That was excellent. Thank you.
Thanks.